Module 11, Vision and Shedding Light on the Eye. So this module is going to deal with all things about the eye. We'll talk about the various structures of the eye. We'll talk about the visual spectrum. We'll talk about how light travels through the eye, from when there's a first physical stimulus to when you actually notice and see something, how that light travels through your eye and then goes up to your brain. So in Module 11, we have two questions. The first question is, what basic processes underlie your sense of vision? So again, this is going to go through how you actually see stimuli that's out there in the environment, the various structures of your eye, and how information has to pass through your eye. And then the second question we're going to answer in this module is how we actually see color. Okay, so let's start out with vision. When we are just talking about basic vision and seeing something, that starts out with light. Light itself is a physical energy that stimulates the eye. So some stimulus out there in the environment will hit your eye, and then there's a whole process that occurs in order for you to actually see that stimulus. Light is a form of electromagnetic radio waves that are out there in the environment. And light is measured in wavelengths. Wavelengths is the distance between two peaks of the light waves. On the next slide, you're going to see the electromagnetic spectrum, and I'll point out what we mean by these wavelengths. The visual spectrum is the range of wavelengths that is visible for us to see. You know, the electromagnetic spectrum is this whole length of wavelengths, but there are only certain wavelengths that occur in order for we as humans to be able to see them. So what happens is when the stimulus hits the eye, your eyes convert light to a form that is used by the neurons in your eye. And that serves as messengers to then travel up to your brain so your brain can see what that message is and figure it out so that you actually recognize it. And that's where the perception aspect occurs. This is a picture of the electromagnetic spectrum. And this is actually on page 95 in your textbook. You can see that all of these are radio waves. So, you know, here we have at 10 nanometers, right? We have FM radio waves, and then we have TV radio waves. We have AM radio waves. We have AC electricity, right? Here at the other end, we have the gamma rays, the X-rays. So if you're having a test done and you have X-rays done, ultraviolet waves. And then you can see there's this very small section along the electromagnetic spectrum from about 400 nanometers to about 700, maybe 800 nanometers. That is the visual spectrum. Within those nanometers is where we can actually perceive information. So this figure, this is actually on page 96, and it says here at the bottom, although human vision is far more complicated than the most sophisticated camera that you see, in some ways it's very similar to that which we see in photography. So the automatic lighting system of a traditional non-digital camera and the human eye dilates to let in more light and contracts to block out light. The same thing that your regular camera used to do. Most people today have digital cameras, but for those who do not have digital cameras, it works the same way. So you can see at this box in the upper right, a camera's lens focuses the inverted image on the film. You can see this inverted tree in the film, and the same way that in your eye, the lens focuses the image on the retina. so it also inverts it the same way that a camera would. So we're going to start talking about the various structures of the eye. So when light, a physical stimulus, first enters your eye, it travels through the cornea. Now the cornea is the outer part of your eye. It's transparent, it's almost like this clear film, and it is there in place to protect your eye. If you would take your finger, if it's clean, and touch your eyeball, what you are actually touching is the cornea. The cornea, again, is there to protect your eye. So you could scratch your cornea, you could tear your cornea. Not that you would want to do these because it's very painful, but you could and it would heal itself. It's there so that your eyeball is actually not injured. Now, because the cornea is curved, it bends or refracts the light as light passes through. The primary role is to focus the light more sharply. As you can see here at the bottom, this is as light travels through your eye. So we said that light, physical property, physical stimulus, first hits the cornea, right? That outer transparent film on your eye. It then travels to the retina, or to the iris, excuse me. It then travels to the iris, which is the colored part of your eye. In the center of the iris is your pupil. That's that dark circle in the center of the iris. Now, the pupil size can change depending on the amount of light that is entering your eye. It changes by the muscles of the iris. So in bright light, the pupil is going to get smaller to allow less light in. In dim or darkened light, your pupil is going to get larger in order to allow more light in so that you can see better in that situation. You may have at some point in time gone to the eye doctor and had an eye test, and they put those drops into your eye so that they could see through your eye. What those drops actually do is they paralyze the muscles of the iris so that your pupil cannot move. And so once your pupil gets really large, it stays in that enhanced state so that they can look through your eye. That liquid that they put, those drops that they put into your eye, that's going to take a couple of hours to wear off. So you may have noticed when you left the eye doctor, you went outside and it was very bright outside. Normally, your pupil would shrink in size. But what those drops did is they paralyzed the muscles of the iris, and now your pupil cannot shrink. And so it is very big, allowing all this light into your eye, and it becomes very uncomfortable. Okay, so once light passes through the cornea to the iris to the pupil, the next place that it wants to go is through the lens. So once light passes through the pupil, it enters the lens. Now, the lens is very important, not as though the other ones aren't, but the lens is important because it bends the rays of light so that they are properly focused on the back of the eye. Now, some of you may wear corrective lenses. Perhaps you wear glasses or you wear contact lenses. That is because your lens needs some help in order to focus the information that you're seeing. And this is accommodation. It refers to the process by which the lens changes its thickness in order to focus the light. So some of you may be nearsighted, which means that you can't see objects far away, or you may be farsighted, which means that you can't see objects up close. I know that sounds opposite, but that's the way that it is. If you're nearsighted, you can't see objects from far away. And if you're farsighted, you can't see objects up close. So what is the issue with the lens? Well, what happens is that the lens becomes flatter when viewing distant objects, and it becomes rounder when looking at closer objects. And so the contact lenses or the glasses that you wear help with that accommodation because your lens is not becoming flat enough or it's not becoming round enough. And so it's going to help with accommodation, and it's going to help with focusing the information coming into your eye. So from the cornea to the iris to the pupil to the lens. Now we reach the back of your eye. The back of your eye is the retina. The retina is the part of your eye that actually converts that electromagnetic energy that we saw in the electromagnetic spectrum earlier. It converts that energy of light into an electrical impulse, and then that information is transmitted to your brain. So once that light stimulus reaches the back of your eye to the retina, it's going to convert that energy from electromagnetic energy to an electrical impulse, and then it's going to travel to your brain. Now, the retina itself has two kinds of light-sensitive receptors. One of those is rods. Rods are what allow you to see in black and white. So rods are definitely helpful in low, dark, or dim light. Cones are what allow you to see in color. So they are really helpful in bright light. So the retina has two types of light-sensitive cells, the rods, which are very sensitive to light, right? So they're more helpful whenever it's dark or dim outside, and cones allow you to see in color. So they're really helpful in bright light. Now, an image reaches the retina upside down. If you remember from a few slides ago, whenever we had that picture of the camera and we had the picture of the eye, and we said that whenever it reaches the inside part of the camera, it's flipped upside down, and the same thing whenever it reaches the back of the eye, when it reaches the retina, it's flipped upside down. However, your brain automatically interprets the information in its original upright position. When that image reaches the retina, it's upside down, but your brain will interpret it as though it is right side up. Now, this is figure 3. This is on page 97 in your textbook, and this figure has a lot of information, right? Here is the back, here's a picture of the eye, right? First off, and you have these light waves that are coming in. You can see the cornea, you can see the lens, here's the retina at the back of your eye, right? The optic nerve, which is what this is. Here's the back of your eye on the right-hand side. You can see your rods and you can see your cones, which are the receptor cells for the retina, correct? And then you see there are two other kinds of cells, the bipolar cells and the ganglion cells. These are layers of connecting neurons that come out of the rods and cones. So if you read along the bottom, it says light entering the eye travels through the ganglion and bipolar cells and strikes the light-sensitive rods and cones located at the back of the eye. And then the rods and cones transmit those nerve impulses to the brain by way of the bipolar and ganglion cells. Now, there are other things that occur in order for this information on its way to reaching the retina. We have different types of vision. Your peripheral vision is when you see objects that are outside of the main center of focus, right? So if you use your peripheral vision, that is what you can see on the sides of your eye. If you look straight ahead at your screen right now, and you take your right hand and you put it to the right side of your head, and without moving your eyes, you can see your right hand, that is your peripheral vision. Dark adaptation. This is the phenomenon of adjusting to dim light after being in really bright light. So if you've ever gotten to a movie theater late and the lights are already dimmed and the movie has already started, you go from really bright light out in the lobby to going inside the movie theater and everything's really dark. It takes your eyes time to adjust or to adapt to that dim light. The same thing happens if you leave the movie theater and go back out into the lobby. Then you have light adaptation. This is where your eyes have to adapt to the bright light after being in a dark place. Sometimes this happens if you've ever watched a film in class, and after the film is over, your teacher turns on the light and you're like, "Ooh, that's so bright." Your eyes take time to adapt to that now bright light. So let's talk about how the message goes from your eye up to your brain. Remember we said you have these two nerve cells, right? Or these neuron cells, rods and cones in the retina. The rods contain what is called rhodopsin. It is a reddish purple protein, and its composition changes chemically when it's energized by light. Your rods contain rhodopsin, which is this reddish purple protein, and the composition of that rhodopsin changes when it's energized by light. Stimulation of the rods and cones in the eye trigger a neural response that is then transmitted to other nerve cells in the retina. And these other nerve cells are the bipolar cells and the ganglion cells. So once the rods and cones are stimulated, they're going to trigger this neural impulse that is then transmitted to the bipolar cells and the ganglion cells. From there, information goes to the optic nerve. The optic nerve is a bundle of ganglion axons that are what actually carry your information to the brain. So information comes into your right visual field, goes through your eye to the retina, to the optic nerve, and it travels that information up to your brain. Now remember, it crosses over and goes to the opposite side of the brain. So information coming into your right visual field will go to the left hemisphere, and information coming in from your left visual field will go to the right hemisphere. You may remember that whenever in a module previously, and we were talking about split brain patients. This optic nerve passes through the retina where there are no rods and cones, therefore creating a blind spot. The optic nerve travels through the retina where there are no optic nerve, or where there are no rods and cones, and therefore it creates this blind spot. This figure 5 you can see on page 99 in your textbook, and it says at the bottom, if you read at the bottom, the optic nerve coming from the eye splits at the optic chiasm. The image to the person's right eye is sent to the left hemisphere in the brain, and images coming in from the left eye switch to the right hemisphere of the brain. And that crossing from the left and right visual field crosses at the optic chiasm. And you can see in this picture, right, here's the optic chiasm at the back where the information is switching from each visual field. It's then going by a certain optic tract. So you have an optic tract on the left side of your brain and an optic tract on your right side of your brain. And from there, the information is going to go to the visual association area in your brain. Your brain's going to figure out what that image is, and then you're actually going to perceive it. So what happens when you're processing the visual message? The processing, right? This image has now traveled through your eye and it goes up to your brain. This message goes up to your brain. And that processing takes place in the visual cortex of the brain, specifically the occipital lobe. If you remember previously when we were talking about the various lobes of your brain, that was back in chapter 3. So the processing takes place in the visual cortex of the brain. We have what are called feature detectors. These are very specialized neurons that are activated only by visual stimuli having specific features, such as, a ball is round, so there's going to be a feature detector for round objects. If you look at, let's say, a baby block, that is square, that's going to have a specific feature detector in your brain so that your brain can figure out what it is that you are looking at. There are different parts of the brain that together they process these nerve impulses according to the attributes of a specific image. Again, whether it's round, whether it's square, what color it is, what size it is. And there are specific parts of the brain that specialize in different kinds of stimuli. There are special parts of the brain that specialize in different kinds of stimuli. All right, so let's finish this module, module 11, by talking about color vision and color blindness. The 7 million color spectrum so that we can see all these various colors. A person with normal color vision is capable of distinguishing at least 7 million different colors. So if you have normal color vision, you should be able to tell the difference between 7 million different colors. However, if you are an individual with not normal color vision, you may be what is called colorblind. And that's where you have quite limited ability to perceive color. True color blindness is where you do not see any color. However, there are different types of color blindness that you could actually see, and we're going to talk about those in the next slide. Individuals who do not have normal color vision and have some sort of color blindness, that's approximately 7% of men and 0.4% of women. So color blindness is much more common in men than what it is in women. So let's try and explain these various parts of color blindness, or what exactly we mean by experiencing color. We have this trichromatic theory of color vision. The trichromatic theory of color vision is, again, tri meaning 3, right? So there are 3 kinds of cones in the retina. Each of those cones responds to a specific range of wavelengths. So if you go back to that previous slide where you saw the radio waves, right? And there was that very small section from 400 nanometers to about 700 nanometers. And that was your visual field for color vision. Each color has a certain wavelength from 400 nanometers to 700 nanometers. You have blue-violet, which is on the left-hand side. So those are the smaller numbers of nanometers. You have green, which is in the middle. So that's mid-range between 400 and 700 nanometers. And then you have the yellow and red. So that would be on the right-hand side of the spectrum. So that's going to be your higher numbers of nanometers. So again, it goes from 400 to 700 nanometers. Blue and violet is on the left, green is in the center, and yellow and red is on the right. We also have what is called the, oh, sorry, I forgot one part here. This is actually challenged by what is called an after image phenomenon. And that's where the colors in the after image differ from those that you originally saw. You can see this on page 102 in your textbook, and it is actually figure 7. I don't have it here in front of you, but it is a very well-known after image example where you are supposed to stare at the black dot in the middle of the flag. Let's say you stare at it for about 1 minute. And then when you look away, what you will actually see is the American flag of red, white, and blue. Well, this comes about because of what is called the opponent process theory of color vision. The opponent process theory of color vision. This is where the receptor cells for color are linked in pairs, and they work in opposition to each other. So if you go back and you look at figure 7, right, you see green and black stripes. So green and red, or green and blue, and yellow and red. I think it's green and red and yellow and blue are actually the opponent processes. I said that wrong. I had to think about it a minute. But green and red are the opponents, and yellow and blue are the opponents. So while one is activated, the other is not activated. And then whenever you look away, you actually see the opposite. That's that after image. But again, you have this opponent process theory of vision. So receptor cells for colors are linked in pairs that are working in opposition to each other. So while one is activated, the other one then is not activated.